Okay, so we now go back. We go to slide. Okay, so this is an example of a furan. Furan. It has 4 carbons. Carbon 1, carbon 2, carbon 3, carbon 4. This is a pyran. Carbon 1, 2, 3, 4, 5. So pyranose formation of monosaccharides. So we have a pyranose. This has 6 carbons. Carbon 1, 2, 3, 4, 5, 6. So it's going to form a cyclical structure, which is called, this is the open chain. This is the open chain. So we are forming a cyclic structure, and this one will lead to the formation of alpha. So this is you can see that the OH is pointing down, so it's alpha. But if the OH is pointing up, then it is what? Beta. Do you understand? So carbon 1, this is carbon 1. This is carbon 1. So if the OH is pointing down, it is alpha. If the OH is pointing up, it is beta. So this is our first one is pyranose formation. We are now forming furanose. So in the end, we get what? Alpha. The OH is pointing down, so we get alpha D-fructofuranose. So this is a fructose, a ketone. You can see there is no H here. There is rather R group and alkyl group, CH2OH. So it's a ketose. Okay, so let's go. Let's go and do some small reading. Disaccharide. A disaccharide is the major form. Is the sugar form when 2 monosaccharides are joined by glycosidic linkage. There exist 3 common examples, which are sucrose, lactose, and maltose. So sucrose is made up of what? Sucrose is made up of what?
Sir, please, which one?
Sucrose is made up of what? What sugars?
Sucrose.
What sugars make up? I said disaccharides have 2 monosaccharides that are joined together by a bond which is called glycosidic bond or glycosidic linkage. Linkage is the same as bond. So examples are sucrose, lactose, and maltose. So I'm saying sucrose. Sucrose, for sucrose.
Sir, please, glucose and fructose.
Glucose and fructose. Clap for her. Clap for her, or? Okay. Okay, it's okay. Then the lactose contains what?
The lactose and glucose.
The lactose and what?
Glucose.
Clap for her. Who is this? Annabelle.
Sir.
God bless you, Annabelle. You didn't disgrace me. Like the way Sibdo is disgracing me. Maltose contains what?
Glucose and glucose.
Clap for Annabelle. Next slide. Annabelle, if I like you are closer to me, I would have given you 50 CD. Okay, so disaccharides. So you see, I said disaccharides are sugars that contain 2 monosaccharide units. So we have here a monosaccharide. So this OH plus OH will react, and when they react, they will give off water to form O. So you see water is given out to form O. So this bond will be what? O. O is like the way O is here. This O was there because water was removed. O was there before water was, so O has been, so water has been removed, so we get what? O. Do you understand? Or I should go to whiteboard?
Sir, please go to the whiteboard.
Okay. So let's see. Okay, so let's see. If you have OH plus OH, what do we get? We get what? Water.
Water.
Plus O. Do you understand?
Like
Do you understand? So water was removed.
Yes, sir.
And so this is a dehydration reaction. Any reaction that is dehydration means what? You remove what? Water. And so this is a dehydration reaction. So water was removed. So it was left with only what? O. So let's go back. So when we remove the water, it's left with what? Only O. Do you understand?
yes, sir.
Do you all understand?
Yes, sir.
Yes, sir.
So this bond, the bond over here highlighted in green, is called a glycosidic bond. So that's the name, glycosidic bond. That is what? A bond between sugar units is called glycosidic bond. Okay, so let's see. Condensation and hydrolysis of disaccharides. So you see, there is one OH here, there is one OH here. So if you condense this, condensation means that you are putting together two or more compounds together, or two or more molecules together. So if you join this one and this one, you get what? A glycosidic bond. We call it condensation. But if you want to get this structure back, then you add water to get it back. So let's go to whiteboard. So over here we have this structure. If we want to get what we had at first, then what do we do? This is O, so we add water to it. H2O. When we add water to it, we end up getting OH plus what? OH. Do you understand? Let's count. The oxygens are what? 1, 2. The oxygens are what? 1, 2. The hydrogens are what? 2. The hydrogens are 1, 2. Do you understand?
Yes, sir.
Do you all understand?
Yes, sir.
Okay, thank you. So let's go. So you see, this is a glycosidic bond, and it's glucose and fructose, and Annabelle said it's called sucrose, and I said you should clap for her, but some of the guys in the class is paining them that why should a woman get answer and they are not getting, so they didn't clap. I saw them. Let's go to the next. So this is maltose, contain glucose and glucose, and this is the glycosidic bond. But you can see water has been removed. Okay. This is lactose, contain galactose and glucose. You can see the glycosidic bond. This is the bond. So let us name glycosidic bonds. So this is very interesting. Let's go back. Let me ask this question. Let me ask this question. Who can number this carbon for me? Okay, let's go back. Okay. Okay, so you can see this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. Is it true? Is it true? Hello?
Sir.
This is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. Is it true?
Yes.
Yes.
Eh?
Yes, sir.
You don't understand. Okay.
No, sir.
This is carbon.
That is fine.
Do you know that this is carbon 1? This is carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. Do you understand?
Yes, sir.
Oh, but
Yes.
this one, if you say you don't understand,
Yes, sir.
sir, if you say you don't understand, then I don't know the kind of human being are you. Because we have even written the numbers, the numbers are there. The numbers are there. Number 1, number 2, number 3, number 4, number 5, number 6, and still you are saying no, sir. Eh?
The man of the work.
The number they work, eh? Do you understand?
Yes, sir.
So this is carbon 1. The OH group, where is it pointing?
Up.
Apples.
Upwards. Okay.
Yes, sir.
Prosper. The OH group over here, where is it pointing?
Down.
Down. Okay, clap for Prosper has done well. Prosper, the OH group over here, where is it pointing?
Down.
Okay. Prosper, the OH group over here, where is it pointing? Prosper, where is it pointing?
Stop, stop, stop.
What carbon number is this?
About 3.
Okay, so let's go back. So we are going to name this one as what? Alpha. So this one is called alpha G glucopyranose. Do you understand why it is called alpha?
Yes, sir.
Yes, sir.
Okay, so let's go. Okay, so let's come here. This is this one and this one. This is alpha. So this is what? This sugar unit. Where is it pointing? The OH on the anomeric carbon. Where is it pointing?
Apples.
Upwards. So is it alpha or beta?
Beta.
Okay. Then what carbon number is this?
6.
Carbon 6. Okay. So this is going to be called beta 1 6. Beta 1 6 glycosidic bond. This is glycosidic bond. This is the spelling glycosidic bond. So this is called beta 1 6 glycosidic bond. Do you understand?
Yes, sir.
Okay.
Yes, sir.
Okay. Then this carbon 1 of this first molecule, where is it pointing?
Apples.
So is it alpha or beta?
Beta.
Beta.
Beta. And this is carbon what?
Carbon 4. Carbon 4. 4.
Carbon 4. So it is going to be called beta. It's going to be called beta what? Beta
1, 4.
1 4. The 1 is for what?
But sir, the carbon is pointing downwards.
Which one? Here.
The other carbon, the one at the right side.
Yes. So you see, so you have asked a very important question. It's pointing downwards, but that is not what we are using as a reference point. What we are using as a reference point is the anomeric carbon or carbon 1. You understand? Do you understand?
Yes, sir.
Yes, sir.
So this beta is in reference to carbon 1. carbon 1 and not carbon 4. So we don't care what happens to carbon 4. You saw that when we were here, I said when we were here, you saw that when we were here, I said beta 1 6. Over here, did you ask me whether the OH is pointing up or down? Did you ask me that?
No, sir.
No, sir.
Yes, because we are not looking at the OH over there. There is no, the OH over there is around here. We are not looking at that. We are looking at the carbon that it is bonding to. So let's go to the third one. This is what? Okay, let's go here. This one, is it alpha or beta?
It's
Alpha.
Alpha. So alpha 1 what? Alpha 1.
Alpha 1.
Alpha 1 what?
2.
Very good. You are very, very okay. So now when I put, can't I cover this thing so that they will not see? Okay. Okay. So name this one for me. This last one. Fast. Fast. Name it fast.
beta 1.
Beta 1 what?
Where's the winter?
Beta 1 what?
Beta 1 1.
Beta 1 1. You saw it. You saw it. So you see, so this one, do you know how we name this one? Because it is on the anomeric carbon, your question now, your answer now comes, or your question. This is beta 1, and this is also what? This is beta because it's pointing up. But this is an anomeric carbon, This is an anomeric carbon, and it is pointing down,
Yes.
And this is carbon 1. So it becomes beta what?
1 alpha 1.
Alpha 1. Do you understand it now?
No, sir.
No, sir, because here we are looking at the bond is between 2 anomeric carbons. 2 carbon 1. This is the same as what? This. This is carbon 1. And it is pointing down, so it is alpha. This is carbon 1 of the same molecule. If it is glucose, of the same glucose. And this is pointing up. So you say beta 1, and this one is what? Alpha 1. You saw over here is pointing up, and this is also pointing up. So it is beta 1 1. Beta 1, beta 1. But if it is not in relation to the anomeric carbon, but any other carbon, then we don't bring either the beta or the alpha there. So whenever we talk about either beta or alpha, it is about the anomeric carbon.
Yes, sir.
Any other carbon is not important, so we don't show whether it is alpha or beta. But if it's at the anomeric carbon, then it is alpha 1, alpha 1. But this one is not the anomeric carbon, so we don't bring any alpha or beta. Do you understand?
Yes, sir.
Okay, so let's go back. So when you go and practice this. Okay, so we have the disaccharides of physiological importance. So we see when you have milk may contain what? May occur in urine during pregnancy. So Annabelle and Co, when you are pregnant, milk can occur in your urine. Lactose will occur in your urine. It's a normal physiological process. Then sucrose, if you are not pregnant and you have lactose in your urine, in your toilet, you have lactose in your toilet and other things, they call it what? Lactose intolerance.
The lactose in the breast.
That one is a normal thing. That's what you drank before you became a man and you are talking ho like that. I'm talking about lactose in the urine. When the women are pregnant, you can see lactose in their urine, and it's normal. So, but if it is if your body is not able to digest lactose, we call it lactose intolerance. Like people who have ulcer and others, they have lactose intolerance. Others also have genetic or congenital lactose intolerance. We also have people who cannot digest sucrose, so they have, because they don't have the enzyme called sucrase. So you can be seeing them having diarrhea anytime they take sugarcane or they take sorghum or pineapple or carrot. Oligosaccharides, like I said, are a type of carbohydrates formed when 3 to 10 simple sugars are linked together through glycosidic bonds. Oligosaccharides can have many functions, including cell recognition and cell binding. So these are oligosaccharides. You can see the glycosidic bonds. Oligo means that they have between 2 and 10. So this one, between 2 or 3 to 10 sugar units. So this one has what? How many sugar units are here? How many monosaccharides are here? 3, okay. Abigail, how many monosaccharides are here?
4.
Okay, Prosper, how many glycosidic bonds are here?
10.
How many glycosidic bonds are here?
8.
Glycosidic bond.
1. There are 4.
Who can tell me the answer? They are what?
3.
Okay. Oligosaccharides. Examples are fructose oligosaccharides, which are found in many vegetables, consist of short chain. This one, go and read them. Then we have mannane oligosaccharides, to go and read them. This is a structure of oligosaccharides. Okay, so for oligosaccharides of therapeutic importance, when oligosaccharides are consumed, the undigested portion serves as food for intestinal microflora. What are intestinal microfloras? What are intestinal microfloras? This word, intestinal microfloras.
So they are the microorganisms found in the intestines.
And what do they do?
And the internal they are natural microorganisms that are found in the intestines of an organism.
What do they do?
They consume all digested.
No, they are microorganisms. They don't cause any disease. They are, most of the time, they are commensals. They are there. They are not causing any disease, but they don't allow a pathogenic microorganisms to attack you. Do you understand? Hello? So, like I said, when oligosaccharides are consumed, the undigested portion serves as food for the intestinal microflora. So somebody was explaining microflora. You know, microflora, they are, when you look at your armpit or axillary region, or your pubic region, where your genitals are, you see that always that place is very, very wet. Why is it wet? Because, you know, that place is where the organisms live most. Most microorganisms love that place. That's where they sleep, they play, they cook, they do everything. And it is prone to infections also. That's why, you know, microorganisms, they like moisture. Where there is moisture, they love it. But over there, God has positioned some microorganisms there. They will not cause any sickness. And they too, when they are somewhere, the pathogenic organisms cannot grow there. They can be there all right, but they cannot multiply. Why? Because those organisms are there. The normal microflora, they are there. They are living there. They are not causing any disease, but they are there. So if the pathogenic or disease causing organism come there, they will not beat you, but they will not give you food to eat. They will not give you water. They will not give you sunlight, because they say what they have is not enough for them. So they will not give you. You said what? Hello? So what they have is not enough for them. So they will not allow any other microorganisms to come and grow there. You can be there all right, but they will not give you food to eat. So you yourself, you will die or you will live there. So that is normal microbial flora. They are all over this thing. Some are in the intestines, some are in the pubic region. They will not allow any organism to grow there. But if those women who like using Dettol and camel antiseptics and other things to be washing their vagina and other things, when you do that, you are doing something we call douching. So you will douche the place. Then this organism, you will kill all this normal microflora. Then when a disease causing microorganisms comes there, it will see that the normal microflora who will compete with it for food, oxygen, nutrients, and light and other things, they are dead or they are not plenty. So now the disease causing organism will grow. It will grow and outgrow the normal microbial flora. Then when they outgrow the normal microbial flora, then you will see that you will start having rashes, diarrhea and other things there, because you have taken a lot of antibiotics and other things and you have killed them. So now the disease causing organism, when they come there, they will now cause a clinical disease. So depending on the type of oligosaccharide, different bacterial groups are stimulated or suppressed. So these are examples. Let's look at polysaccharides. Polysaccharides are long chains of monosaccharides linked by glycosidic bonds. Three major polysaccharides include starch, glycogen, and cellulose. Three important polysaccharides include starch, glycogen, and cellulose. So you can see this is a polysaccharide, more than 10 sugar units, and they are joined together by glycosidic bonds. Homopolysaccharide means that homo means that the same. So for instance, if Annabelle were to be sextuplet, or Annabelle was giving birth and 6 children came and they are identical, we say that Annabelle sextuplet or Homo Annabelle, meaning that they are the same. When you look at them, they resemble each other. So these are the same sugars. Homopolysaccharide. Another homopolysaccharide is here. But if there is glucose is here, fructose is here, galactose is here, and they are formed a long chain, they are heteropolysaccharides. Heteropolysaccharides. And it is an unbranched chain. It's a long straight chain. This is also unbranched, but this one has branch. This one has branch. Functions of polysaccharides. Polysaccharides are structure. They provide mechanical stability to cells, organs, and organisms. Water-binding polysaccharides are strongly hydrated and prevent cells and tissues from drying out. So, example of a water-binding polysaccharide is starch. It can absorb water. Storage polysaccharides, starch and glycogen. Structure polysaccharides, cellulose and chitin. Cellulose is found in plant, and chitin is found in what? I've forgotten where chitin is found, so find out. Water binding polysaccharides are dextrans and what? Agarose. So when you go to the microbiology lab, they will teach you agar, what an agar is. Agar is what we grow the microorganisms on, and it is starch. It is a polysaccharide. It absorbs a lot of water, so the organisms love to grow on it. Then dextran or dextrin is what we mix with when you go to hospital and some people are on drip. They use dextrose because it's able to absorb a lot of water. So they add the dextrose or sugar to the drip, so that somebody who has done an operation or is having coma will depend on the dextrose sugar for food, because in that condition they can't eat any food. Functions of polysaccharides. Even though these complex carbohydrates are not very digestible, they may comprise important dietary elements for humans. They are called dietary fibers. These carbohydrates enhance digestion among other benefits. So you go and read on this. Okay, this is very important. Polysaccharides can be combined with proteins or lipids to form other biological molecules. For example, peptidoglycans are found in cell walls of bacterial cells. Gram-negative bacterial cells. They have what? Peptidoglycan chains, which are a combination of proteins. It's a combination of proteins and polysaccharides, and are found in a cell wall of certain bacteria. So this, when you hear pepti is what? Peptide, peptide or protein, and glycan is what? Glucose or carbohydrate. So we can also combine glucose and lipids to form glycolipids, which is a combination of polysaccharides and lipids, and this also constitutes cell membrane. Let's look at examples. So we have modified monosaccharides. This is a normal glucose. If you add an acetyl group, this is an acetyl group, CH3 C double bond O, is called acetate. You remember the acetic acid that I draw, 2 carbons. This is acetate, and this is an amino group, so we call it N amino group, so we say N acetal. This is an acetal group. Let me draw an acetal group on the whiteboard. CH3. Oh, anye samaba. So CH3 C this is an acetal group. So we have an amino group here. So amino has what? NH. Then it is also bonded to what? Another H. That's ammonia. Anytime you hear amino, is ammonia. Then we have double bond here, a lone pair of electron here. So this is the amino group. Amino group. What is another amino group? They form what? They form amines, and amides. They form amines and amides. And this is an acetal group. Acetal group. The acetal group can be what? R C O RCO. So RCO. Okay. Acetal. Okay, so let's go back to the whiteboard and go and finish up the lecture. Okay, let's go back to the lecture. So this is N-acetal D-glucose. This is a D-glucose. This is also N-acetal D-galactosamine. So this is the N and this is the acetal group. Acetal group is CH3CO, then bonded to something. Do you understand? Do you understand?
Yes, sir.
Hey, are you the only one around? I'm seeing everybody is around.
Yes, sir.
Okay.
Yes, sir.
So let's see. We want to modify glucose again. So this is acidic monosaccharides. So you can see that this is a normal glucose, but on the carbon 6, instead of the CH2, on the carbon 6, on the carbon 6, on the carbon 6, most of the time you see CH2OH. This is carbon 6. So we have what? So this is C. Let me clean this. CH2. So this is carbon 6. So instead of the CH2OH on the carbon 6, we have what? COOH or COO minus. Do you understand? So anytime you see COO, anytime you see COOH, hey, the COOH, it is called carboxylic acid. Carboxylic acid. Carboxylic what? Acid. Do you understand?
Yes, sir.
Do you all understand? Eh?
Yes, sir.
Okay.
Yes, sir.
So this is what? A carbonate. So the proton has left. The proton is not there. This is the proton on the whiteboard. So this one is a proton. So when it leaves with the positive, it will leave something like this. So this is a carbonate. Hey, carbonate, let's take it like that. So this is a carbonate. So this is carboxylic acid. So that is, we call this one, this is a levorotatory iduronate acid. So you can see that the COOH is pointing down. COAH on carbon 5 is pointing down. But this one, this is carbon 5 and this is carbon 6. So the carbon 6 is part of the COO minus. So we call it D-glucuronic acid. But iduronate acid is rather, it has what? 5 carbons. 1, 2, 3, 4, 5. And it has 6 carbons. Just that the COH is pointing down instead of it pointing up here. The carbon 6 is pointing down. The same way we have N-acetal neuraminic acid. This is a neuraminic acid. So we say neuraminic. This whole strata is called N-acetal neuraminic acid. This is vaccination. We don't need to talk about this slide because I will teach you this in immunology. So by this we have come to the end of the class.